Gelato or Nah? Phage Discovery by Rigoberto Guerrero, Caden Thompson, and Christopher Lopez. Introduction and materials and methods presented by Chris Lopez.
Introduction. Phages are essential to the biological processes of both eukaryotes and prokaryotes. In order to come up with possible solutions in phage treatments, phages have to first be discovered and analyzed. In this experiment, 2 bacteriophages were extracted, grown, and analyzed. One came from Collin College parking lot, one came from a lab member's house. The host used for the bacteriophage infection was Microbacterium foliorum, which is a non-pathogenic species of the Microbacterium genus. The basic protocol used for extraction, purification, and analysis is described in Figure 1, which follows the protocols found in the Phage Discovery Guide.
First, we collected our environmental samples, like you heard from Chris, from the Collin College parking lot and the lab member's house. Then, we purified our plaque assays, diluted them, and created our webbed plates, which resulted in these from Gelato and Nah. And then we made our lysates, and from those lysates we used them to make our enzyme digest, which resulted in these pictures of Nah and Gelato. And then we sent them off, our lysates, to get them photographed, and this is Gelato and this is Nah. And now presenting the conclusion is Rigoberto.
So out of our soil samples, we were only able to extract 2 phages. As you can see, C is Gelato and D is Nah. There wasn't really a scientific process to naming these. They were just chosen at random. As for Gelato, which is figure 2C, the capsid size was 67, which is a little on the shorter side. However, the tail was 142, which is on the longer side, compared to Nah, which the tail length was 110, which is really on the short side. So what figure A and B show us is our enzyme digest. Figure A is Nah and figure B is Gelato. With our Nah enzyme digest, we didn't have really dark bands, which is, you could say, a problem, but not really, because we did have some light bands, which are outlined in red. What you can also see are some white, opaque circles. Those are anomalies in a gel. You really don't see that. What I would suggest for our gel is running it again, just in case we missed something or we did something wrong. However, I'd say at the end it was actually successful, since we did have bands, which showed us that there was phage DNA and that the enzymes did digest that DNA, showing us that there was actually some phage in there. As for Gelato, we had way better results. As you can see, the enzymes NSP and SAC2 had way better results with darker bands at different decisions, showing us that it did digest the DNA and that there was a lot of DNA present in order for the enzymes to work and digest the DNA. So overall, I'd say we were able to run the enzyme restrictions. However, I would just suggest rerunning Nah, just to further clarify and to solidify that we did have adequate DNA. Since most of our tests were successful, such as getting the pictures and cutting up the DNA with the restriction enzymes, the last step would really be to sequence the DNA in order to figure out whether we discovered a new phage or the phage has already been discovered.
Thank you to Dr. Twishel for helping us through the entire project.
And a special thanks to the University of North Texas for allowing our images of our phages to be captured.
Also, thank you to Collin College and the biotechnology department for supplying us with the materials necessary for this project.